So it's my pleasure now to spend the next 40 minutes or so talking about the biochemistry of sterols, bile acids, and oxy sterols. So a couple of, or three useful resources which are worth looking at the end of my talk. At the top, I've got a link to lectures which are actually on the Lipid Maps website. There's some very nice lectures by Ed Dennis from the University of California discussing the biochemistry of lipids, and two of those are on sterols. So I recommend those as a watch. And then I've got two YouTube videos given by, first one's given by Goldstein and Brown, where they describe their work on cholesterol, which led to their winning of the Nobel Prize in 1985. And the last link is to another YouTube video, this time only by Mike Brown, where he describes how you go about winning a Nobel Prize. So that's some videos to watch, and at the end I've got two slides at the end which give a list of references which really go with this talk. So this talk is really about the lipid maps category of sterol lipids, and we're going to be looking at the lipid maps classes of sterols and bile acids and derivatives. So I'm going to be talking really about the biochemistry in mammalian systems, really, because that's my bias. So an outline of the talk: we'll have a short introduction to sterols in general. Then we'll look at the regulation of cellular cholesterol. We'll then move on to look at cholesterol metabolism, and then we'll specifically look at bile acids and oxy sterols. So let's start with the introduction. So all sterols are based on this cyclopentanoperhydrophenanthrene skeleton. So this is the cyclopentanoperhydrophenanthrene skeleton. It's this four-membered ring system. Now, sterols, they're present in all eukaryotes and some prokaryotes. And unsurprisingly, cholesterol was the first of all the sterols to be discovered. It was first isolated in 1915 from gallstones by Michel Chevreul, and the chemical structure was determined in 1888 by Friedrich Reitz. However, it wasn't really until the X-ray structure was determined by John Bernal that the correct chemical structure of cholesterol was determined. So that was determined in 1932 by Wieland and Dane and Rosheim and King. So that really came from Bernal's work, really, of providing the X-ray crystal structure. So cholesterol, it's the most important of all the sterols. So at the top, I've got the structure of cholesterol. So you can see there are 27 carbons, and we number them from left to right. There are 4 rings, the A, B, C, and D rings. And we have a side chain which has got 8 carbons in it, and a C8 side chain. So cholesterol is a mostly hydrophobic molecule. It's got essentially a planar structure, so the rings are in a plane. However, it's got a 3-beta hydroxy group, which gives us some polarity, and that's pointing upwards. So in animal cells, the levels of cholesterol are around 10 femtomoles per cell, and most of that cholesterol is actually present in the plasma membrane. So it seems that about 60 to 90% of cellular cholesterol is present in the plasma membrane. That corresponds to about 45 mole% of total lipids. So there's a lot of cholesterol in each animal cell. So all mammalian cells have the capacity to synthesize cholesterol from scratch from acetyl-CoA, and there are over 20 enzymes involved in the synthesis of cholesterol. So acetyl-CoA is converted through a number of steps to squalene 2,3 epoxide, which is this molecule, in a pathway which we call the mevalonate pathway, and then squalene 2,3 epoxide is converted to lanosterol in an incredible reaction, really. So the enzyme lanosterol synthase actually cyclizes this squalene molecule and sort of simultaneously makes 4 carbon-carbon bonds. So I think that's an incredible reaction. So once we got lanosterol, there are a number of other further enzymatic reactions going on, and we ultimately get cholesterol. Plants can also synthesize sterols. They have a mevalonate pathway as well, but plants can convert squalene 2,3 epoxide into this molecule, cycloartinol, which has got this extra 3-membered ring in it. However, in this talk, really, I'm only going to talk about mammalian sterols, which are cholesterol related. Okay, that's cholesterol synthesis. Well, cholesterol also needs to be metabolized, and the first step of all cholesterol metabolism is the formation of an oxysterol. So oxysterols are a family of oxidized forms of cholesterol. So oxysterols are metabolized further. They're metabolized to steroids like progesterone, cortisol, estradiol, and testosterone. But the majority of cholesterol metabolism leads to bile acids. And the common bile acids, the primary bile acids, are cholic acid and chenodeoxycholic acid. A few more words about bile acids. So at the bottom right of my screen here, I show you the structure of cholic acid. So you can see it's a little bit like cholesterol, and it's got the ring system. However, the side chain has been shortened from 8 carbons down to 5 carbons. And we've got this carboxylic acid group introduced. So that's why they're called bile acids. We've also got additional hydroxylations in cholic acid at carbon 12 and at carbon 7. The double bond that was present in the B ring of cholesterol has been reduced, and we've had an inversion of the stereochemistry of the hydroxyl group at position 3. So actually quite a lot of chemistry has gone on when we go from cholesterol metabolizing it into bile acids. So in general, bile acids are synthesized primarily in the liver. However, some bile acid synthesis, at least the early steps of bile acid biosynthesis, can occur extrahepatically. So, for example, actually, some early steps of bile acid synthesis can occur in the lung and in the brain. Bile acids are secreted from the gallbladder into the gut, and in the gut, their job is to aid the absorption of fats in the intestine. They're antimicrobial, which is an added bonus. And amazingly, really, 90% of the bile acids that are secreted into the intestine are reabsorbed and recycled via the enterohepatic system. So they're reabsorbed in the intestine and are transported back to the liver. And those bile acids that are not reabsorbed, they're excreted in the feces. So we have a mechanism of eliminating cholesterol from the body. What's also very interesting is that many intermediates in the pathways from cholesterol to bile acids are actually biologically active in their own right. So this is actually what's really interested me in bile acid biosynthesis pathways. Okay, that's the introduction. Now let's get into the meat and drink of sterol biochemistry. So first of all, let's look at the regulation of cellular cholesterol. How are the levels of cholesterol regulated in cells? So this was really worked out by Mike Brown and Joe Goldstein in Dallas, Texas. And for their work on cholesterol, they actually won the Nobel Prize in 1985. So their win was for the discoveries concerning the regulation of cholesterol metabolism. And I'll try and explain in the next few slides a little bit about how cholesterol is regulated. So if you want to hear it from the experts, then click on this link and watch the YouTube video. Okay, so if we look at cholesterol synthesis, on the right-hand part of the slide, I've got an abbreviated version of the cholesterol biosynthesis pathway. Well, basically, we start with acetyl-CoA and we end up with cholesterol, and there are a number of steps in between, and there are over 20 enzymes which catalyze these various steps when we go from acetyl-CoA to cholesterol. One step that you might be familiar with is the conversion of HMG-CoA to mevalonate. This is the step which is regulated by HMG-CoA reductase, and HMG-CoA reductase is the enzyme which statins inhibit. So, as I said, there are more than 20 enzymes involved in the synthesis of cholesterol from acetyl-CoA, and the expression of all the genes coding for all the enzymes in this pathway are regulated by a master transcription factor called SREBP2, or sterol regulatory element binding protein. So this master transcription factors regulates the expression of all the genes of all the enzymes in the cholesterol biosynthesis pathway. So each of the genes of these enzymes has a sterol regulatory element, and the SREBP binds to the sterol regulatory element and activates target gene transcription. The LDL receptor, which takes up LDL from the bloodstream into cells, that also has a sterol response element and is also regulated by SREBP2. So SREBP2 regulates the expression of enzymes of the cholesterol biosynthesis pathway. It also regulates the uptake of cholesterol via the LDL receptor. So let's look at how this SREBP2 sterol regulatory element binding protein 2 works. Well, SREBP2 is synthesized in the endoplasmic reticulum. And when it's synthesized, it immediately binds to another protein called SCAP. SCAP stands for SREBP cleavage activating protein. And the job of SCAP is to transport SREBP from the endoplasmic reticulum to the Golgi. So SREBP is transported to the Golgi. When it gets to the Golgi, it's processed by two enzymes. So SREBP is actually activated, and we end up with a active form of SREBP, which then translocates to the nucleus and activates those target genes for the expression of enzymes of the cholesterol biosynthesis pathway and the LDL receptor. So this is how SREBP, this master transcription factor works. So what happens when a cell has low levels of cholesterol? SREBP is transported by SCAP to the Golgi. In the Golgi, SREBP2 is processed to its active form. The active form of SREBP2 activates gene transcriptions for enzymes of the cholesterol synthesis pathway and for the LDL receptor. So this increases cholesterol synthesis, and it also increases the uptake of cholesterol from the bloodstream via the LDL receptor. So this is how the SREBP pathway works. So a few words about the LDL receptor. So this is also regulated by the SREBPs. And how the LDL receptor takes up LDL cholesterol, this was also worked out by Goldstein and Brown. And they described how LDL is taken up by, how LDL particles is taken up by LDL receptors through a process they termed receptor-mediated endocytosis. So what happens? We have LDL particles in the bloodstream. So these LDL particles will contain lipids, they'll contain an apolipoprotein, and they'll contain cholesterol esters. So the LDL particles bind to LDL receptors. So the LDL particle becomes bound to the LDL receptor, and the LDL particle becomes internalized into an endosome via this process of receptor-mediated endocytosis. So now we've got our LDL particle containing these cholesterol esters in the endosome. The endosome then combines with the lysosome, and in the lysosome, proteins from LDL are broken down to amino acids, and cholesterol esters become hydrolyzed. So cholesterol becomes available to the cell. So the cholesterol is then exported out of the lysosomes into membranes of the cell, including the endoplasmic reticulum. So the cell is fed with more cholesterol. So what happens now when the cell has got sufficient cholesterol? It's got enough cholesterol. Well, what happens is that excess cholesterol, which is present in the endoplasmic reticulum, binds to the protein SCAP. This causes a conformational change in SCAP, and SCAP then binds to a ER resident protein called INSIG. So INSIG is really an anchor protein. So SCAP binds to INSIG, and SCAP becomes anchored in the endoplasmic reticulum. So SCAP can no longer transport SREBP from the endoplasmic reticulum to the Golgi. So if SREBP can't get to the Golgi, it can't be processed into its active form as a transcription factor. So it's essentially, cholesterol biosynthesis is turned off as we've inactivated the SREBP transcription factor. So cholesterol biosynthesis is turned off. Also, expression of the LDL receptor is turned off. So the amount of cholesterol in the cell no longer continues to increase. So I've just written that out in a few sentences. So when cholesterol in the endoplasmic reticulum exceeds about 5 mol% of ER lipids, then cholesterol in the ER binds to SCAP. SCAP binds to INSIG. INSIG then tethers the SCAP-SREBP complex in the ER. There's no transport of SREBP2 to the Golgi. There's no activation of SREBP. So there's no activation of the enzymes of cholesterol synthesis, and there's no upregulation of the LDL receptor expression. So this is actually, Goldstein and Brown termed this convergent inhibition, how cholesterol will actually regulate its own biosynthesis. In fact, this schema from Goldstein and Brown really describes things really rather nicely. When the cell has got enough cholesterol, when the endoplasmic reticulum has got enough cholesterol, then the enzymes of the cholesterol biosynthesis pathway are downregulated. So there's a reduction in the amount of HMG-CoA being produced. There's a reduction in the amount of LDL receptors being produced. There's also an increase in esterification of cholesterol to remove any further excess cholesterol. So this is how cholesterol regulates its own synthesis and uptake. So the the schema from Goldstein Brown that I just showed here. This has now been updated a little bit in recent years, and it seems that cholesterol from LDL particles, which goes to the lysosome, it seems that that cholesterol, it first goes to the plasma membrane before it goes to the endoplasmic reticulum. In Goldstein and Brown's scheme, they had the cholesterol going directly from the lysosome to the ER. However, it seems now that there's very good evidence that cholesterol from LDL particles goes from the lysosome into the plasma membrane, and then from the plasma membrane into the ER, where cholesterol biosynthesis is regulated. So this gives me an opportunity to introduce you to a topic which describes pools of accessible and sequestered cholesterol in the plasma membrane. So this is something that is now becoming, it's clear this is now becoming increasingly important. This fact there are different pools of cholesterol in the plasma membrane. So first of all, it's important to recognize that most of the cellular cholesterol, 60 to 90%, is actually present in the plasma membrane, where it makes up about 45% of the total lipids in the plasma membrane. Now, the majority of the plasma membrane cholesterol is sequestered by sphingomyelin or other membrane factors. So the majority of the plasma membrane is sequestered. So that means it's effectively it's imprisoned. Then there's a small signaling pool of plasma membrane cholesterol, which can be transported from the plasma membrane to the ER to signal SREB processing. This is called accessible cholesterol. So that there are at least two pools of cholesterol in the plasma membrane. There's the accessible cholesterol pool, which is a small pool, and this is the regulatory pool. While there's a much larger pool, which is essentially the imprisoned pool, which is imprisoned by sphingomyelin and other membrane factors. So the next question really is, how are changes in levels of accessible cholesterol actually monitored by the endoplasmic reticulum? So this is recently been explained by groups from Japan and the US. And if we look at the figure on the far left, so we've got plasma membrane on the outside, and we've got branches of the endoplasmic reticulum coming towards the plasma membrane. So in the endoplasmic reticulum, we have proteins called Asta proteins. Now these Asta proteins, they contain a membrane binding domain and a cholesterol transfer domain. Now when the levels of accessible cholesterol in the plasma membrane increase, then the Asta proteins will bind to the plasma membrane, and they will then transfer accessible cholesterol from the plasma membrane into the endoplasmic reticulum. This cholesterol can then be monitored and will affect the SREB pre-processing. So the endoplasmic reticulum monitors the accessible cholesterol in the plasma membrane through these Asta proteins. So this provides a connection between cholesterol levels in the plasma membrane and cholesterol levels in the ER. And of course, it's in the ER where all the SREBP machinery is present. Okay, so that's, I've hopefully given you an idea of how how cholesterol is maintained, is regulated in the cell. I'll just finally have a slide describing accessible cholesterol and cholesterol homeostasis. So in a summary, we can say the majority of cellular cholesterol is in the plasma membrane, and a small proportion of which is accessible cholesterol. The ER monitors levels of accessible cholesterol in the plasma membrane via Asta proteins transporting accessible cholesterol to the ER. Elevated levels of accessible cholesterol in the plasma membrane lead to elevated levels of ER cholesterol, and that leads to inhibition of the SREBP processing. This reduces cholesterol synthesis, and it also reduces expression of the LDL receptor. Okay, let's now change track, and let's move from cholesterol synthesis to cholesterol metabolism. Well, the major route of all cholesterol metabolism is metabolism to bile acids. So there are two major pathways of bile acid biosynthesis. There's the neutral pathway on the left and the acidic pathway on the right. So the neutral pathway, which is present, drawn here against the blue background, this pathway proceeds in the liver. And on the first enzyme of the neutral pathway is CYP7A1, and that introduces 7-alpha hydroxy group into cholesterol to form 7-alpha hydroxycholesterol. This first step is the rate determining step of the neutral pathway of bile acid biosynthesis. We then have very many other steps in which the side chain of cholesterol is shortened, a carboxylic acid group is introduced, we have extra hydroxyl groups, and we have an inversion of stereochemistry at C3. So the neutral pathway is the dominant pathway of bile acid biosynthesis, and this proceeds exclusively in the liver. So we also have the acidic pathway of bile acid biosynthesis, and the early steps of the acidic pathway can occur extrahepatically. For example, early steps can occur in the lung, and also early steps can occur in the brain. Now, the first step of the acidic pathway of bile acid biosynthesis is introduction of a 27 hydroxy group by CYP27A1. CYP27A1 will then oxidize carbon 27 from a primary alcohol to a carboxylic acid group. So the carboxylic acid is introduced into the second step of the acidic pathway of bile acid biosynthesis. Hence, that's why it's called the acidic pathway. We then have side chain shortening and hydroxylation, and we ultimately get chenodeoxycholic acid. So cholic acid is the primary product of the neutral pathway. Chenodeoxycholic acid is the primary product of the acidic pathway. So I suppose the next question is, how is bile acid biosynthesis regulated? We've seen that we can form bile acids. How is this process regulated? Well, first of all, cholesterol is transported from the periphery to the liver by a process of reverse cholesterol transport. So excess cholesterol in the body can be taken back to the liver for conversion into bile acids. The bile acids themselves, which are secreted into the intestine, they're also returned from the intestine via the enterohepatic system back to the liver. So we've got bile acids being returned, and we've also got bile acids which are going to be synthesized. So I've said that the dominant route for cholesterol metabolism, certainly in adults, is the neutral pathway. And the neutral pathway is regulated by activation of the FXR nuclear receptor in the liver. So FXR, that's a nuclear receptor, And nuclear receptors, they're ligand activated transcription factors. So actually bile acids are the ligands for FXR. So what actually happens is bile acids bind to LXR, activate transcription of target genes, and these target genes decrease the production of bile acids in the liver and increase bile acid secretion from the liver. So this protects the liver from toxic effects of elevated bile acids. So essentially we've got a negative feedback mechanism here. So elevated levels of bile acids in liver cells will turn off their bile acid, turn off their synthesis, and increase their secretion. So I've got a cartoon here describing how the FXR nuclear receptor works in the hepatocyte. So bile acids in the hepatocyte, if we've got elevated levels of bile acids in the hepatocyte, they will be imported into the nucleus via a nuclear pore. Now in the nucleus, we've got FXR, this nuclear receptor, and it sits on the promoter of its target genes. Normally, FXR forms a heterodimer with another nuclear receptor called RXR, and heterodimer is normally in the off position, and it's got a co-repressor attached to it. However, bile acids come into the nucleus and they bind to FXR. This causes the replacement of a co-repressor by a co-activator, and the result is we get activation of target gene transcription. Now, one of the target genes that transcribed is SHP, small heterodimer partner. SHP mRNA is exported out of the nucleus to the ribosome, and SHP protein is formed. Now, SHP protein inhibits CYP7A1, and CYP7A1 is the enzyme that catalyzes the first step of the neutral pathway of bile acid biosynthesis, which is the rate determining step. So the expression of the enzyme of the rate determining step is turned off, so bile acid biosynthesis is turned off. So this is actually how bile acid synthesis is regulated in the hepatocyte. Okay, in the last 10 minutes or so, let me now finally talk about oxysterols. So oxysterols, they're oxidized forms of cholesterol. They're a large family of molecules. So we've already met two oxysterols. So 7-alpha hydroxycholesterol, that first member of the neutral pathway, that's an oxysterol. So is 27 hydroxycholesterol, that first member of the acidic pathway. And we find that different cells and different tissues make different patterns of oxysterols. So macrophages when activated make high levels of 25 hydroxycholesterol. So the hydroxy group is at position 25. Neurons in the brain make 24 hydroxycholesterol, hydroxy groups at position 24. Then the placenta and the adrenal cortex, that makes 22 hydroxycholesterol. So different tissues and different cells makes different oxysterols, and these oxysterols can be metabolized further to further oxysterols, making we end up with a really big family of molecules. So, is this important? Well, let's discuss a little bit more about oxysterols. So they're a family of oxidized forms of cholesterol. They're intermediates in bile acid and also steroid hormone biosynthesis. An important point about oxysterols is that they cross cell membranes much quicker than cholesterol. So they can exert their biological activities much quicker than cholesterol. So they're biologically active. They regulate cholesterol synthesis. They activate ACAT, which is that enzyme which esterifies cholesterol. They also activate nuclear receptors. So they actually are ligands to Lxr, the estrogen receptor, and the glucocorticoid receptor. They're also ligands to G protein coupled receptors, for example, GPR183s. That's also known as EBI2. That's a nuclear receptor important in the immune system. They also activate another nuclear receptor called smoothened, which is important in hedgehog signaling, which is important in development. They're also modulators of neurotransmitter receptors, such as the NMDA receptor. So this family of oxysterols, which contains very many different molecules, the family has lots of biological activities. So of the oxysterols that I think probably are the most important, these are what we call the side chain oxysterols, where the added oxygen functionality is in the side chain. So like 27-hydroxycholesterol, 25, 24, and 22. These are all side chain oxysterols. So these oxysterols, they modulate SREBP processing, and they're also ligands to the Lx receptor. So I've already shown you how cholesterol modulates the processing of SREBP2. Side chain oxysterols also modulate the processing of SREBP2, but in a slightly different manner. So instead of binding to SCAP, oxysterols directly bind to the ER resident protein called INSIG. So an oxysterol will bind to INSIG. This causes a conformational change in INSIG. So again, INSIG will bind to SCAP, and again, SCAP will be tethered in the endoplasmic reticulum. So if SCAP's tethered in the endoplasmic reticulum, it can't take SREBP to the Golgi. SREBP can't be processed, so it can't act as a transcription factor for cholesterol biosynthesis. So side chain oxysterols also regulate cholesterol biosynthesis. And I guess the general feeling is that they're probably important for the fine tuning of cholesterol biosynthesis in the cell. Oxysterols also activate the nuclear receptor Lxr. So Lxr, it's expressed in liver, brain, and many tissues. One of the most important target, or some of the most important target genes of Lxr are the transporters ABCA1 and ABCG1. These are cholesterol transporters, and they will transport cholesterol across membranes. So if Lxr is activated by oxysterols, you get increase in expression of these transporters, and these will transport excess cholesterol out of the cell. So the net result of activation of the Lxr is cholesterol export from cells. An indirect effect of activation of the Lxr receptors is also degradation of the LDL receptor. So overall, activation of Lxrs will reduce cellular cholesterol. So just a cartoon showing you how oxysterols function. So oxysterols, they will bind to the Lxr nuclear receptor, which is sitting on its sitting on the promoter of target genes. Co-repressors are replaced by co-activators. We'll have target gene expression. So for example, in this case, expression of ABCA1. ABCA1 protein will be generated and cholesterol can be exported from the cell. So the overall effect of the oxysterol is to reduce the cellular cholesterol. I'll just have one last slide where I'll just talk about one particular oxysterol, which is called 25-hydroxycholesterol. And this is probably the most well-studied of all the oxysterols, because for many years it's been commercially available from Sigma. So this is the oxysterol that people will test. Endogenously, it's formed by the enzyme cholesterol 25-hydroxy cholesterol 25-hydroxylase, or CH25H, so CH25H. Levels of 25-hydroxycholesterol in cells and in the bloodstream are normally rather low. So really, until recently, although 25-hydroxycholesterol has got lots of activities in vitro, its levels in vivo, in biology, are normally very low. So it's really been questionable whether really it's an oxysterol of any importance. However, in about 2009, it was shown that the expression of CH25H and the levels of 25-hydroxycholesterol are hugely elevated in response to bacterial and viral attack. And it seems now absolutely conclusive that 25-hydroxycholesterol is really important in the defense of the immune system against the invaders. And this defense almost certainly goes through activation of the LXR and also inhibiting of SREBP processing, and also activation of ACAT. So it seems that 25-hydroxycholesterol, it's really important for defending the body against bacteria. It's also important for defending the body against viruses as well. And if I had another half an hour, I would show you some more slides on that. But I think I've been going on for 40 minutes now, so I guess it's time for me to finish. Okay, so just at the end, I've got a couple of slides which give you references to what I've been talking about.